Hello students, so we are here to again start our topic Mendel experiment. As you all know, in previous videos we have did that Mendel selected the 7 traits, and these traits were always seeds, flower, pods in the stem. These traits were like seed shape, cotyledons, color and flower color, pod color and pod form, and the stem position and its size. So on these basis, he studied and did the experiments in the 3 steps. He took any one of the characters from these 7 traits and did his experiment by following 3 steps. The first step was like you can say he took the trait like size of the plant. If he raised tall plants again and again from the same seeds, and he observed the pure tall plants he get in the next generation, and from there he took the pure plants. He did it by doing self-pollination. By doing again and again self-pollination, the purity he get in the plants, and he obtained one character for the one seed particular. Second step was he did the second step, the production of the first generation by hybridization. From these pure plants, when he cross-pollinated them, and he obtained by taking 2 pure plants of one particular trait, like we were saying that tall character was taken, so plant size character was taken, and the plants like one is tall, one was dwarf, and they get cross fertilized, and cross pollination was done between 2 pure parents, and he got the F1 generation or the first generation by the hybridization. So he obtained by doing it cross pollination. And you know, if you want to cross pollinate from one pure plant to another pure plant, then you have to follow 3 conditions, like emasculation, bagging, and tagging. Emasculation, you have to first remove the anther of one flower which you want to take as a female parent. So you have to remove the anther in the bud condition and cover it with a polypal, so that no other flower, no other pollen grain can pollinate it. And after that you have to put a tag, name tag, that you have pollinated it by which flower. So by doing these steps, you can produce first generation. His third step was raising of the second generation. So he raised second generation plants by doing self-pollination under controlled conditions, means they will not be pollinated by any other flower, they will be pollinated by itself, by its own anther, and it was done by using a bag, poly bag, so that another flower cannot pollinate it. So Mendel experiment can be concluded as a 3-step process, like one is the selection of pure plant, second was the production of first generation, and third was the raising of the second generation. So first he did the experiment by using one trait only. So we said that it is called inheritance of one gene, that is known as monohybrid cross. Means if you consider one trait, the cross or the hybridization process for doing only one trait, that is called monohybrid cross, and Mendel first did the monohybrid cross only for the one trait, single trait, and individually for other traits he did the same experiment by using only one trait at one time. So first of all, he did this. He took the character as plant or the stem height. Stem height, as you all know in the previous lecture, that the traits were in the contrasting pair. Means they have only two options. Stem height में दो ही option थे, tall and dwarf. So as we know, three steps were taken by the Mendel. First he obtained the pure parents. Those, one was taken as male and another was taken as a female part, female flower. And these were the parents like pure, tall and the pure dwarf. There is no mixing of character here. So there were, these were there. So here are these are our parents, tall and short, short means dwarf. Tall plant and dwarf plant, they were closed, hybridized. By doing cross pollination, we get the F1 generation. This was our F1 generation, and we obtained all plants were tall. From here, you see that there is only one parent character was shown in the F1 generation. Next, then he did the self-fertilization. In self-fertilization, he crossed same all tall plants with each other. And he obtained here 3 plants were tall and 1 was short. Now, if there was no character in F1 generation, no short character was there, but parents में वो short character था. So that character is carried from one generation to next generation and was shown only in the third generation, in F2 generation. This was F2 generation. So what he observed, we can conclude that what he observed in this experiment, number one, F1 resemble with either of one parent. Means in F1 generation only one parental characters were shown. Second, F1 represents only one parent, not the other parent. Other parent characters was not shown. Then the self-pollinated F1 plants, what was we obtained? In F2 generation, we obtained again parental combination. If these are 4, out of 4, 1 was there which had a parental combination. So in F2 generation, 1/4 express other parent combination. Other parent character. In F2 generation, other parent character was shown only the 1/4 of the plants. Third, there was no plant of the intermediate height. Tall and short, these two characters don't blend with each other and didn't blend with each other, and there were no blend मिला था, कोई भी मिक्स कैरेक्टर नहीं मिला था. There was only contrasting characters were there. So from these observations, what they concluded? First conclusion was that something was transferred, that was stably transferred from one generation to next generation. That something was called factor by Mendel and later named as gene. Later it was named as gene. Mendel called that factor which was stably expressed, stably transferred from one generation to next generation. Second, these genes which code for a pair of contrasting traits are known as alleles. Gene pair, that is known as allele. Third, one trait expressed in F1 generation that was known as dominant, while other trait which cannot express in F1 generation was recessive. From here, for dominant, capital letter was used. And for recessive, small letter was used. So, from here, if we were doing, or Mendel was doing the experiment on the plant height, so tall character which expressed itself in the F1 generation was represented by the dominant capital letter T, and recessive was expressed by the small letter t. From here, we get that if there is an allele present, they can be same, or the alleles can be have different. So if these are similar, it is known as homozygous. And if it has two different alleles, two different dissimilar alleles, it is known as heterozygous. This can be understood by using Punnett scale. Punnett scale is a graphical representation to get the maximum possibility which combinations can occur from the different gametes. given by Pune, a scientist, British scientist was there. He gave that graphical representation, which is represented here. The gametes, male gamete or the female gamete, are represented on the top border and the left side. So we have possibilities of these two gametes in F1 generation, and these pairs that represent the possible combinations of these gametes. From here also you can see there are the three represent tall character and one only represent dwarf character. The ratio is 3:1, and this ratio was called monohybrid cross ratio. So here is a representation of the Mendel experiment by taking one trait only, and from here we obtained this result like monohybrid cross ratio 3:1. You can conclude it here. Mendel did the experiment in 3 steps by taking the pure parents, by raising F1 generation and by raising F2 generation. Next, he started experiment by taking only one trait, that is called monohybrid cross. For that he took one factor at one time like stem height. He took the one factor stem height as tall and dwarf. He took the pure parents. From these pure parents he obtained F1 generation, and by doing selfing or the self pollination he did obtain the F2 generation, which show the ratio of 3:1. And from here the observation was these 3 observations were there, like F1 resembles the eyes of the one parent, another parent was not showing their character, and F2 generation, in F2 generation 1/4 were expressed there, that parent combination which was not expressed in the F1, and there was no blending of characters of these any trait, two alleles they don't blend with each other. From here he concluded that something was stably transferred from one generation to the next generation, and that something was factor, or the we call now it as a gene. Second, the gene that pair of the gene that have show the character, that is called allele, and that allele can be similar or can be dissimilar. If they are the similar, they are homozygous, and if these are dissimilar, they don't same, so they call the heterozygous condition. One character that which express in the F1 generation of the parent, that is called dominant, and which do not express itself, that is called recessive. And by using Punnett scale you can also represent this cross. These were the parents, pure, tall and pure dwarf. From here the gametes produced are these are the gametes, these are the parents. These gametes combined with each other and give only one combination, that is your F1 generation, and by doing selfing means these were taken as a parent, and they can produce 2 types of the gametes, and this also can produce 2 types of the gametes, and they cross with each other to form different combinations. Like this. From here the 3 were tall and 1 was dwarf, and this also can be obtained as early as by the Punnett scale, like this one. So this was the Mendel experiment on the monohybrid cross, and later in our later videos we will see that what he concluded from, or which laws he gave on the basis of its monohybrid cross. Thank you.